Good. Today we gonna continue talking about morphology of bacteria and being more specific regarding bacterial ultrastructure. So if last week we started discussing about shapes, like spherical shape, rod shape, spiral shape, differences between different types of rod-shaped bacteria, differences between arrangements, and so on. Today we're gonna talk more about ultrastructure of bacteria. That means we'll talk more specifically about morphological differences between bacteria. So, just as you probably know, any bacteria is a microorganism. It has its own morphology. That includes cell wall. It could include some permanent, non-permanent structures, or we also call them essential, non-essential, or we also call them mandatory or optional. Here we can see some examples. For example, cell wall. What means mandatory? It is present in any bacterial cell. Like cell wall, like nucleoid. There is a difference between eukaryotic and prokaryotic cells. In prokaryotic cells we have nucleoid, that means DNA. And in eukaryotic cells we have nucleus. Okay. Here are just few of them, not all of them. Here is a general example of different cell structures, and you can observe that, we can know that not all of them are present in any kind of bacteria. Talking about gram-positive, gram-negative bacteria, we started discussing that during the last class, but still there is a case to repeat. So, if you can see, schematically they look the same, but actually all the time gram-positive is much thicker, gram-negative is much thinner. They look the same just schematically, just in order to see differences between layers. So if we are talking about gram-positive bacteria, it is already drawn here with purple. That's interesting that this one is not drawn with red. It would make more sense, but anyways. You can observe that the first layer is made of more layers of peptidoglycan. Between these layers of peptidoglycan, we have connections made of teichoic and lipoteichoic acids. After that, we have periplasmic space, and after that, cytoplasmic membrane. It is gram-positive bacteria. Gram-negative bacteria, and you can observe 1, 2, 3 layers, 3 different layers in gram-positive bacteria. Meanwhile, gram-negative bacteria has outer membrane. Here and here we have periplasmic space. And after that we have like the one layer of peptidoglycan is situated between 2 layers of periplasmic space, and the last one is cytoplasmic membrane. And as a result, the entire cell wall of the gram-negative bacteria will be much thinner than just layer of peptidoglycan in gram-positive bacteria. So when we apply gram staining, because of strong connections between different layers of peptidoglycan. It makes possible capturing the purple dye, crystal violet, in gram-positive bacteria. And here we don't have any connections, any layers. As a result, it is not able to capture the dye. Yeah. Yes. As a result of gram staining, we will have purple gram-positive and pink pale gram-negative. Talking about chemical composition, basically, similar composition you will observe in most of living organisms. So, almost all the time, water is making the biggest mass of all the microorganisms, all the macroorganisms, animals, vegetation, and so on. On the second place, proteins, nucleic acids, lipids, carbohydrates, and inorganic ions that are responsible on transportation of micro elements, water, and so on. Here I remind about a case. One day, I guess in US, a few guys started making activity for signing petition, in which they declared that according to different studies, there is a substance we should it should be forbidden. So they started asking people to sign a petition for approval to make this substance being forbidden. They told it is dihydrogen oxide, and it is extremely dangerous because it is present in all the pathogens, in all the poisons, in everything that is harmful for us. It is present overall. This is why we should sign it. As a result, a lot of people signed that petition, and nobody understood it was they've been talking about water, because water is everywhere, and there is no process that is possible to happen without water. So actually, pay attention to what you're signing. Here you have. Sure. I'm just passing over the stuff you can read after, and we are talking about the mostly important things now. So actually, what I just passed is largely discussed the entire chemical composition of the cell. Why it is important to know chemical composition? Just one of reasons. It has direct connection to how we can fight with bacteria, because different bacteria can be attacked by different antibiotics through different chemical structures. Talking about essential or mandatory structures, here we have a cell wall, and the function of the cell wall, of course, it is protection. It gives the shape. It gives the transportation of the elements outside the bacterial cell. the function is extremely important and wide. As composition, we already discussed what it is made of. But there is important to keep in mind that cell wall is not present in any bacteria. Here is an example: mycoplasma. It has cytoplasmic membrane. It has cell membrane, but it doesn't have cell wall. And here is the question: is it gram-positive or gram-negative? Huh?
It's neutral.
Neutral. Why?
Because if it doesn't have a membrane, so you cannot talk about anything. It depends on the membrane for the grams, doesn't it? Somewhat?
Not really.
Not really?
Not really.
But it is neutral, isn't it?
It's gram-negative.
It's gram-negative. Give me a reason. What made you thinking about that first?
Gram-negative. I have a.
Do you have any suggestion
No,
here?
I had a hunch, and I searched
For-
gram-negative.
What?
I want to think how I can formulate my thoughts. So,
Because it's a lipid membrane.
I mean,
Wait, is it also going to be like pinkish?
yeah, it's gram-negative, pinkish.
Well, the color is not everything.
Yeah.
Because by any other staining method, a lot of bacteria will get pink or red color. So it is not everything. It is just a result by which we make a conclusion. But still, there is a logic in what you want to explain. But I want you to develop your thoughts.
Yeah.
Why it's negative? Any other opinions?
It have the bigger amount of lipopolysaccharide on its certain outer membrane, and it can induce sepsis, whatever. In case it's.
Is it also harmful?
Of course. Yeah.
It's not the same as mycobacterial. It's not the same. It's not.
Not the same,
Not the same.
for sure.
Mycobacterial.
Maybe because it has a double membrane. Just as we saw a double membrane, an outer membrane and a cytoplasmic membrane.
I mean, if it's gram-negative, it has an outer and an inner membrane.
What is a cell wall?
It's just a polysaccharide like coating around the.
Cell wall.
Cell wall. It's the
The,
peptidoglycan.
the main part of a cell wall is peptidoglycan. So it has only cytoplasmic membrane.
Which is a lipid bilayer. So it'd be the same deal as gram-negative.
When we are talking about gram-positive, gram-negative bacteria, we should understand that it is about, first, about chemical compositions, and second, how do they behave, how these bacteria behaves or interacts with others. So, for sure, it doesn't fit to a classical gram-negative bacteria. For sure, it doesn't fit to a classical gram-positive bacteria, as many other bacteria, because we just classify them in gram-positive, gram-negative, not like exactly the same chemical structure, not like exactly same composition, but how do they act and how do they interact with other organisms, macro and microorganisms, and as a result, how do we treat infections caused by this or that pathogen? As a result, mycoplasma will have structures that are potentially closer to gram-negative because it doesn't have cell wall that is peptidoglycan, and it has other structures characteristic for gram-negative bacteria. As a result, the mostly efficient antibiotics for mycoplasma will be those that are more efficient for gram-negative bacteria. So chemical structure is closer to that one. And here you had a suggestion.
Oh yeah, A, gram-negative.
So in the name you have a suggestion that it is gram-negative, mycoplasma. Cytoplasmic membrane or plasma membrane, it is that membrane that surrounds the cytoplasma. This is why it is called cytoplasmic membrane. So we have a few layers. Excuse me. We have it's kind of one more filter in the bacteria.
I got it confused with the gram variable one.
Gram what?
Variable. The one where it depends on the age of the bacteria if you're after the stain. Gram variable.
Something interesting. Didn't hear about that.
And it's there in the older presentation.
Okay.
I'll show it to you after.
Okay. So.
It's not about this spectrum.
So. Plasma membrane has a structure that you could see in a school program, I guess, right? That is made of lipoproteins, lipids. Here we have lipids with hydrophilic heads and hydrophobic tails. All of them are situated with tails to each other and with heads out and inside the cell. And here we have proteins that are having function of transportation of different substances. And here we have polysaccharides, that are having function of receptors, playing role of receptors. So transportation has place through the cell wall by different ways, like simple diffusion. It is passive and active through proteic pumps that are working independent on presence or absence of some anorganic substances on different edges. As a result, these structures, they help to keep osmotic pressure. If you remember what is osmotic pressure, we can have osmotic pressure, we can have oncotic pressure. Osmotic pressure depends on presence of salts, and oncotic pressure, presence of proteins, right? So the difference of concentration of these salts, the solute, will give higher or lower osmotic pressure. As a result, it will make constriction or enlargement of the cell. This is why it is important to keep isotonic pressure. What means isotonic pressure?
One is the same, equal.
Equal between outer and inner space. Correct. Of course, cytoplasm, ribosomes, nucleoid, all of this function, that's clear. And talking about optional or non-essential structures, capsule. Whenever we are talking about capsule, we are thinking about what?
His outer wall.
The function is protection. What is interesting, not every bacteria that has capsule has it for the during the entire life. Some bacteria are able to start producing capsule in unfavorable conditions. So during the lifetime, if everything is fine, they live without any capsule. After that, they yeah. The function of capsule, of course, is protection, is survival, and namely protection against phagocytosis. What is phagocytosis?
Yeah. It's the digestion of debris,
And foreign substances.
dead
Yes.
cells.
Phagocytes that are present in our immune system, in our body, they have ability to engulf foreign substances, including bacteria. And if bacteria is capsulated, this is how it looks under. Of course. This is how it looks under the microscope. This is bacteria, and this is capsule. So this capsule is made of polysaccharides. Polysaccharides are very neutral chemical substances. Like we have very aggressive acids and bases. Both of them can produce damage. And it doesn't matter is it a base or acid around. You can neutralize bases with acids and acids with bases, but polysaccharides they have that neutral pH that cannot be affected by both of them. This is why, by the way, our mucous membranes are covered. What is mucus?
It's polysaccharide.
Polysaccharide. It also has that neutral pH. This is why our digestion process doesn't produce any harm to our mucous membranes, because in some portions of our digestive tract there is acidic pH, in others there is basic pH, and none of them in normal situation will not produce any harm because of these polysaccharides. Same thing happens here. Digestion, right, is not possible for these polysaccharides because they're neutral, and it is very hard to dissolve them and to destroy the bacterial cell. As a result, bacteria that are able to produce capsule, or encapsulated bacteria, after phagocytosis, like macrophages are engulfing the bacteria, is able to survive, to persist inside of the macrophages. After that, macrophages, they have their own life cycle that finishes after engulfing of foreign substances, like they initiate the process of auto-destroying. And after that, everything that is inside of the macrophages, if it is survived, it is released outside and they are continuing their life cycle. So it is really good protection. And of course, somehow capsule can protect against other environmental factors, but not that much as against phagocytosis. Also, you will find in some places information that capsule has as a secondary function attachment to surfaces. Of course, especially if it is mucous membrane, it will have good affinity to that mucous membrane because of similar chemical structure. But basically, the main function is not attachment. The main function is protection. Another yes.
Does our body still have a way to kill such bacteria?
Sure, because still, except polysaccharides, except capsule, it has different proteic structures. Moreover, every foreign substance that enters our body, and specifically all three, proteins, lipids, and carbohydrates that have foreign origin, they are antigenic for us. The highest level of antigenicity that bothers our immune system will have proteins. This is why, by default, we call all the foreign, all the antigens, we call them proteins, because most of them are they have proteic origin. But still, foreign lipids and foreign carbohydrates, they also have a certain level of antigenicity. Anyways, some proteic structures will have free access to the outer side of bacteria. These bacteria for sure will be detected by our body, and it will produce. Our body will start producing antibodies, but it is another topic. So still, of course, they are not. They are also vulnerable, less vulnerable than other bacteria. They have more protection, but still, of course, against any action we can find a contra-action. Talking about the next one is flagella, and the function of flagellum is motility, is movement. Here we have some examples of different types of flagellum. The first one, the first example, it has only one tail, only one flagellum, and it will be called monotrichus, with the only one flagellum. Also, it can be on one side bunch of flagellum. It will be called lophotrichus. Lopho means bunch, one bunch on one side. We can have bacteria with flagellum on both sides. This one is called amphitrichus. So, as it can have one flagellum on each side and be called amphitrichus, as well it can have two bunches of flagellum, like here, and it will be also called amphitrichus. And all around the bacterial body is peritrichus. But it is not the only example of motile bacteria. One more example of motile bacteria is spirochete, bacteria with a spiral shape. And the difference between such flagellum and flagellum present in spiral-shaped bacteria is that flagellum in spiral-shaped bacteria is called endoflagellum, or also it is called axial filament. And that axial filament is why it is called endoflagellum or axial filament, because actually if you will observe the structure of spiral-shaped bacteria, and if you will ask yourself why it has this shape, there is a structure around which one the bacteria is situated, and that structure is flagellum. So a lot of coils going around the flagellum. This is why it is called axial filament, axis. And why it is called endoflagellum? Because this axial filament is situated between layers of the cell wall. So actually from the outside you cannot see it. And its function is it gives contraction of the entire cell. Like worms. They're moving like worms. So if you will see closer the structure of the flagellum, exoflagellum, like we have here, you will observe that it is a hook that is connected to the basal membrane of the cell wall, and it has a kind of rotor that gives rotation to the entire tail. As a result, it works like an engine.
Like a propeller.
Yes. So it rotates and gives spinning movement to the bacteria. When we are talking about bacteria with exoflagellum, so we can compare exoflagellum with an engine and endoflagellum with a muscle that gives contraction and movement.
Are monotrichous bacteria able to pivot?
Again?
How are they able to pivot if it has like only one flagellum? Like to turn
A little bit louder.
louder? Okay. So if it has only one flagellum, how is it able to like deliberately turn in a certain direction?
Not too much.
Not too much. They
Not
just
too much.
it's convenient?
You know, whenever you are talking about motile bacteria, you should understand it is not about
Precise movement.
it's not about competition, who is faster, and how you arrive, like how fast you get from 0 to 100. You know, it is only about changing your position from place to place in order to survive. So they don't really care about that. They have one flagellum, they move a little bit slower. And it is more complicated for them to move in different direction, of course. They have more, they will move faster, but in the same direction somehow. They have, on two sides, they will be a little bit more motile, a little bit more flexible in space. And of course, this one goes everywhere. So, as a result, you will see that as much bacteria, the Pyretrichus is a leader for sure, because it has all the directions for movement. It will multiply faster. It will occupy the entire space faster. So survives that one who is.
What's one example of Pyretrichus bacteria?
Example?
Yeah.
Intestinal infections, intestinal pathogens, like not just infections. I all the time forget, is it Salmonella or Shigella? Because one of them is non-motile and one of them is motile, and well, Salmonella. And Escherichia. Escherichia Lophotrichus and Salmonella Pyretrichus. And you can observe both of them are situated in the intestinal tract. Okay. Pili. And here the main function is attachment, is adhesion to the surface. Who speaks Romanian?
Yeah.
You know that phrase? To have pili? That person has pili somewhere. You know? Are pili peste tot. No? Okay. Well, it is an expression that means the person has connections, has good relations in the right places, you know? Like you have attachment to everything you need.
That's a good way to remember it.
Yeah. And don't you think that this came from popular expressions to microbiology. It went from microbiology in population. Okay. Because pili in microbiology are older. And still, here we have example Escherichia coli. It has a lot of pili and Lophotrichus bacteria, right? And also Neisseria gonorrhea. Both of them, they have very good attachment to substrates and specifically to mucous membranes. As a result, Escherichia coli, actually it is a normal representative of our intestinal tract, but some serovariants are mutated ones, and they are able to produce harm, and they are able to produce intestinal infections. This is why as soon as they will attach to the surface of the large intestine, they will produce an inflammatory process with all the results. Same thing with Neisseria gonorrhea. It has very strong attachment to mucous membranes. As a result, it produces STI that affects too much genitalia in both men and women, but it's more specifically in men, more symptomatic cases. We have plasmids, and plasmid is extra chromosomal DNA, and they encode a lot of important for bacteria information. For example, information regarding antibiotic resistance. Like it is a database of bacteria that collects information about previous contacts with harmful substances. And as a result, this genetical material that contains information about those antibiotics helps encoding the structure of bacteria in order to change it somehow, to not let antibiotics be efficient against this bacteria. Endospores. We've been discussing about different types of rod-shaped bacteria with spore forming. So a lot of people are thinking that the function of endospore is multiplication. It is not so. Multiplication is function for fungal spores. There are some mushrooms that are multiplying by spore production. And these spores are actually like seeds. They are falling down from the mushroom, and they're starting reproducing in this way. While we are talking about endospores in bacteria, the function is not protection of the bacteria. It makes sense because in order to give protection, you should be outside, not inside. The endospore, is hidden inside of bacteria. So it is carried and protected somehow by bacteria till the moment bacteria dies. Usually such bacteria are really susceptible to the environmental factors. As soon as they get into the environment, they will die. The best example are Clostridial bacteria. Clostridium perfringens, Clostridium botulinum, Clostridium tetani. Since they are strictly anaerobic bacteria, that means they are not able to survive in presence of oxygen, and they are multiplying just in absence of oxygen. As soon as they get in the environment that is full of oxygen, they are dying, and spores remain in soil for a lot of time, even hundreds of years, until they're waiting, until someone will get them and put them into favorable conditions. As a result, they will be able to transform from spore to start germinating, transforming from non-living into living form of cell, and starting multiplying. Of course, the main condition for these very bacteria is absence of oxygen, so they should get into anaerobic conditions. Plus, it should be enough. Yes.
Temperature.
Yes. Water, temperature, pH, all the nutrients, everything that gives to these bacteria possibility to multiply. And the function of endospore is survival of species, protection of genetical material. So it doesn't protect the cell, the bacterial cell itself, finally it dies, but it helps generations to come to survive. Yeah. And here is the recap of the entire mandatory, non-mandatory, or optional, mandatory and optional structures with some examples. So I guess this table is going to be useful for you. Now, all of these structures, since they have different chemical composition, since they have different functions, and since they help us specifically to detect different pathogens, because this is what makes them different. Difference in structure. All of these structures can be highlighted in different ways. If we need to highlight gram-positive versus gram-negative, we have gram staining, that's clear. So if we are interested about cell wall chemical structure, we choose gram staining. Whenever we are interested in cytoplasmic membrane detection, in which case we are interested in detection of cytoplasmic membrane?
Case of gram-negative.
In case of
Gram-negative.
so-called, just as you call them, neutral. You know? When in some cases, cytoplasmic membrane is almost the only thing you can find. In such case, we can apply fluorescence microscopy. Detection and yeah.
Is it usual that you find gram-neutral bacteria?
Usual for whom?
As a microbiologist.
When you talk about microbiologist, you're talking about specifically about me or generally? About me. About me, overall, I'm not finding anything. Because most of my job is talking about that. So unfortunately, and it is our reality, like I should be true. I need to choose to work more on in the lab.
So more research.
Or to make more research and working more with students. And this is the problem because we need a good lab in which we could bring you and show you some things.
Do we have a good lab or not really?
Huh?
Do we have a good lab or not really?
Yes, we do. But we don't have access. A lot of sponsors are coming, and it's just between us, and are saying, "Oh, you need a BSL-4 lab, you need BSL-3 lab for the university, for the department, because it is very important to show how it works, and so on and so on." Of course, let's do it. Let's do it. Where are we gonna do it? Makes more sense to make in the department, no? No. Let's do it in the central building. Okay, who's gonna work there? Oh, we already have a person who gonna work there. Okay, do whatever you want. You know?
So it's in the central building.
Yes, nearby. Nearby.
Not even in the central building.
Yes. And they like, please give us all the information how it should be, all the facilities, all the devices, everything it should be, because it is all for you, it is all for students. Let's do it, let's do it, let's do it. Okay. We provided them with all the materials. They are doing something, we have no idea what they are doing.
They're still doing it?
Mhm.
We're not gonna see a bacteria until 6 years.
No. Actually, I'm okay to not have a direct attachment to such things because who knows?
But it's possible to learn in a way that when you don't have that access, right? Like it's not as that we're gonna get a less.
Yes, that's a problem. That's a problem because somehow you stay behind of progress. For sure. What is good about microbiology specifically is fundamental science that doesn't change too much in ascension. But of course, it could be we can get in an awkward situation like in the process of teaching, discussing. Somebody who actually works in such labs is coming. It is not actually relevant for 10 years anymore, you know. So it can happen.
Yeah.
It can happen. It doesn't mean that it doesn't make sense to discuss it anymore, at least like history. But of course, it makes you sometimes frustrated because of that. Anyways, we should be aware about the fact that basic stuff we're discussing here will never change. What is changing? Some chemical structures of some pathogens and methods of their detection and their treatment. And actually it is about chemical composition, not about method of treatment. It's about chemical composition of new drugs. In ascension, nothing changed. This is why there is no risk I will tell you something that is not relevant 10 years after. Like it could happen with other departments. And I've been a witness of such situations. This is what makes me calm on this point, because I know it is still relevant, I guess, till the end of your life, everything we're going to discuss here. But still, for example, here we're discussing how we highlight by different methods different bacterial structures. And at the same time, we can do it, but we don't really do it because, look, ribosomes. We can detect it through electron microscopy, right?
Yeah.
But we are not doing it on the daily basis for diagnosis. Or we can detect cytoplasmic membrane with fluorescent microscopy or with electron microscopy. We don't really need it for diagnosis. We can detect it. So whenever you are interested, how can you prove it exists? By these methods that actually exist. But we don't really do it because it is not relevant in the framework of the diagnosis and treatment. Exactly. Meanwhile, we have express methods of diagnosis. Rapid tests. Antigen, antibody, PCR, and so on. These are golden standards. On the first place, PCR, and we'll discuss it later. And next, antigen, antibody. Some of them are faster, some of them have higher precision, but all of them, they have their limitations anyways. if you want to find something, if you want to prove it exists, please. Again, you want to detect flagellum. How we detect flagellum? Staining, special staining, Loeffler staining. And you prove the flagellum is present. Meanwhile, according to symptoms and according to antigenic test, you don't really need to stain it and to prove it, because you can find it faster. But there are some techniques that appeared before such express test appeared. So these methods have been situated on the basis of development of microbiology that told us that we need to discover something more about these structures. As a result, like you discompose the entire bacterial cell, you study all the antigenic structures, you produce kits that are able to detect antigens of different structures, you apply this antigen, and you find, you prove presence or absence of some structures. Coming back to the question. Coming back to the question for how often I meet different types of bacteria. Personally, about me, very often I meet only one genus and species of bacteria. Staphylococcus aureus, methicillin-resistant Staphylococcus aureus, MRSA, because it is my PhD title. So, as deeper you are going with your research, don't you think as much you know in a world of microorganisms? Because when you're a student and when you are doing your thesis, your diploma research, you can take a lot. When you take microbiology for residency, sometimes they ask for a diploma, for thesis. Sometimes they don't ask for thesis. From year to year it differs. You need to go in a more narrow direction. When you are coming to your PhD, even more narrow, because you should be aware about the fact that micro world is that numerous. We probably never find all the species, all the types of bacteria, and we actually don't really need it. We should be aware about issues, about local issues. Why in different, for example, vaccination? Why do we have in different countries different list of mandatory immunization program? Because in different countries are circulating different pathogens. They're endemic for specific regions. So microbiologists from Moldova will differ from microbiologists from Uganda, that will differ from microbiologists from Paris, that will differ from microbiologists from USA. You know? Just because they have different needs and different targets. And what is very interesting about such labs we have but don't have. We had a scientific event on which have been invited a chief of the scientific lab, and here please make difference. There are clinical labs and there are scientific labs, different stuff. Clinical lab, you're working just with pathogens we take from patients. Scientific lab, you work with everything you want. And he says, we have everything. We actually have everything. Do you want to produce some tests, antigen, antibody tests for the mostly rare infection in the world? We have it. We've been working with Ebola. We've been working with Zika. We've been working with anything you ask.
Here?
Yes, no. There.
Uganda.
No. In Slovenia.
Slovenia.
Yeah. And our clinical staff is asking, does it make sense? It is very expensive. Does it make sense? Because these bacteria and these viruses are not circulating in your region. Does it make sense? This is extremely expensive. And he says, the mostly beautiful thing about scientific lab, you're not asking about expenses. You're not thinking about expenses. You're thinking about the result. That's it. That is actually beautiful from this point of view, but extremely dangerous from another point of view. Why do you need it?
Dangerous in a way that, like, why? Because you're bringing it.
Because. A lot of questions.
Mm-hmm.
A lot of questions. After that, you ask, all of those outbreaks are going before or after?
Yeah.
And nobody tells you. Before pandemic, I would laugh on such ideas. Now, I'm just asking myself, how possible is it? And still, I asked that person. Like you've been showing a lot of graphics with incidents of COVID during pandemic. You've been showing, look, here it goes up. Here we put restrictions on circulation. It goes down. After that we reduce restrictions. It goes up. After that we put restrictions and we enforce people wearing masks. It goes down. After that, we don't make any restrictions, it goes up. After that, we don't make we don't ask people wearing masks, it goes down. After that, we ask people wearing masks, it goes up. And it seems nobody observed that. And I'm asking him, that I hate any form of conspiracy theories. But what you're showing here makes me to give you a question. Does it make sense or not? And is there something beyond all the epidemiological activities we apply to the population that makes incidents going up and down? Because sometimes you see connection between restrictions, and sometimes you don't see any connection. So here is my question: does it make sense or not? And why we are working so? His reply were awesome. He told, I would be careful with such questions. I told, okay. After that, he also replied something, but I don't remember. You know, like my grandfather used to tell me, don't listen to anyone and don't trust anyone. After that, he told something else, but I didn't listen and I didn't trust him anymore. So, this is our reality. Of course, it doesn't cancel the fact all of these pathogens exist. And whenever I'm asked, is it true it was invented in the lab? Who knows, maybe.
But it's still there.
10 years ago I would say, come on. Today I say, I don't know. But it doesn't make any sense. Okay, if I find out the answer, what it will change? Absolutely nothing. So, things I cannot confirm or deny and show scientifically it is so or it is not, I don't know. Go and prove. If you can prove it, okay. If you cannot, it doesn't make sense to discuss it. The facts we can prove and things we can find are shown here, and we have a deal with them on a daily basis. This is why it is much more important to pay attention to things you meet actually very often, but not to look for something extremely rare, because you can open Pandora box. And this is why I prefer being that microbiologist who is aware about everything or about most of things. But I'm working with methicillin-resistant Staphylococcus aureus that is present all over the world, is present in any hospital. We have a lot of work with that. And we will have during 100 years for sure with this pathogen. This is extremely important pathogen, an extremely ordinary pathogen. And nothing is that complicated about that pathogen except its ability of producing resistance for different drugs. I'm gonna work on that. Probably it doesn't bring that much money, but I don't care. Because yes, it is very important to be okay with what you have. Very, very important. And if you can afford your existing and existing of your family, and still you have a little bit extra, fine. Why not? Why not? Finally, if you will get much more than others.
There's a point when you're not gonna be more happy. There's a point where
Moreover,
you're gonna be over the moon, and after that it's gonna be okay.
Yes. Moreover, there is no point when it is enough on the one side. There is no point, because your hunger will increase with your income. And on the second, on the other side, life becomes boring when you don't have a goal.
Yeah.
Who used to play video games with cheat codes? Is it interesting?
At least I tried. But
Is it interesting?
It depends.
At first,
When you can make unlimited bank account, when you're immortal, when you everything, and nobody can do anything to you.
Usually it's done after you have everything.
That's not the first.
It doesn't.
first,
First is fun. Wow, nobody can do anything to me. But it doesn't last for too long. Same thing with money. Same thing with money. When you have enough to cover everything you have immediately, right here, right now, it is not interesting anymore. So you will never be happy by having a lot. You will be happy by achieving your goals, time after time. This is why it is better to be successful but ordinary employee or chief or whatever.
Because it's more stable also, usually.
Yeah. By the way, keep in mind, people who get high fast enough fall fast enough. As longer you will go up, as larger will be your base. Okay, moving forward. Staining technique we already discussed. So here is just a recap for that. You remember, first of all, we put crystal violet, about 1 minute waiting. And as a result, if we stop it here, both gram positive, gram negative will be purple. I love this projector. Yeah. After that, we put some iodine that makes strong connection between crystal violet, peptidoglycan from gram-positive bacteria. And after that, we apply decolorizer. Usually it is alcohol or acetone. And it will wash out all the gram-negative bacteria. And here will be intermediate result. And finally, we put safranin, the red dye. And in the end, we just wash it out with water, without decolorizer. Otherwise, we'll get again this result. Since we apply water, water is not able to penetrate that much inside of the cell. It just washes it from the outside. As a result, gram-positive and gram-negative bacteria, you will see difference. Students very often are misusing gram staining with simple stainings because it is mostly common. Most of all, you hear about gram staining. This is what makes you think it is simple staining. But simple staining is staining with only one dye by only one step. You apply a dye, you wash it out, you microscope. That's it. One step, one dye. Everything that has more than one step, more than one dye, more than one chemical applied for the smear, it is complex staining. So gram staining is the mostly common complex staining ever. Next one, acid-fast staining. And here is an example when I ask you, gram-positive bacteria, is it red or purple? Or blue maybe, or what? Gram-positive. Gram-positive is purple, gram-negative is pink or red. Wrong.
They're not that.
Gram staining.
Because you didn't answer, you didn't listen to my question.
You're asking the bacteria itself.
And
They're color.
Yeah.
Yeah, they're clear, yeah.
They only
the
become-
question was, is gram-positive bacteria is purple or red or... And basically, it is colorless. Now, when you get such questions, you should ask me back. Initially or after staining? Okay, after staining. After which method? After which technique? And then I ask you, okay, Seal-Nielsen technique. Is gram-positive bacteria red or blue maybe?
We use methylene blue, so maybe blue.
So maybe blue. Or maybe red. This is the problem that when you look under the microscope, you can see some red and some purple bacteria. Oh, this one is gram-positive, this one is gram-negative. And other time you look under the microscope, oh, this one is red and this one is blue. What's that? You get confused, right? So, after we discuss Sill-Nielsen staining, you will get the point. Sill-Nielsen staining technique, or acid-fast. This one is by author, this one is by function. Acid-fast, that is acid-resistant.
We have two types, heated and cold, yes? We have two types of acid-fast staining, heated and cold.
How you apply the cold one?
I have no clue, to be honest.
Me too.
The heated one. I saw the cold one, I was like, nothing.
Me either.
Yeah.
So, staying here. Sill-Nielsen staining is the heated one. Which is the idea? Acid-fast bacteria, that is Mycobacterium, and here you keep in mind, bacterium is rod shape, non-spore forming, gram-positive bacteria. By default, you already know it is gram-positive. Of course, you don't know if the patient has Mycobacterium or Staphylococcus or whatever, because different pathogens can produce
Same symptom.
catarrhal symptoms. And you should find out, is it bronchitis, is it pneumonia, is it classical pneumonia, or it is tuberculosis. How you find it out? You make gram staining and you see nothing. Let's make Sill-Nielsen staining. Because acid-fast bacteria, acid-resistant bacteria, that is Mycobacterium. Why it is called Mycobacterium? Because except the entire chemical structure that is characteristic for gram-positive bacteria, Mycobacterium has coating, extra coating made of mycolic acids that are actually wax. This is why it is resistant to acids, it is resistant to alcohol, and any other chemical substances. And if you apply simple, like ordinary method of staining, for example, gram staining, this waxy membrane will not let any dye penetrate. It doesn't matter it's gram positive or gram negative. Any dye will not be able to penetrate this cell wall because of this waxy membrane. This is why after applying the first one is carbolfuchsin, you apply the first dye. After that, we heat it.
5 minutes?
No.
2-3 times?
Seconds. Few seconds. How it works? You take a slide, you put it on the fire, and you take it out. Put it on the fire, taking it out, and you're waiting for steams. But before it will start boiling. Because if it will start boiling, if you will lose that moment, it is a moment. So when you lose that moment and you boil it, you destroy the bacteria, you will see nothing. This is why you need just to wait for whitish steams. You take it out, you let it getting cool. You can repeat it one more time just to be sure and take it out. That's it. What happens on that moment? The waxy layer melts. The dye penetrates inside. After that, waxy layer comes back solid. And when you apply alcohol or when you apply, usually, I don't know why they put it here specifically alcohol. Of course you can use it, but usually it is 5% solution of sulfuric acid. Just to make sure 100% because it is more aggressive. So it will wash out all the carbolfuchsin from all the non-acid resistant bacteria. and only acid-resistant bacteria will stay red. And after that, the last step, we apply methylene blue. This time we don't heat anymore anything. We just put here methylene blue, just to evidence to show evidence or difference between acid-fast and non-acid-fast bacteria. And here is my question: which one is gram-positive and which one is gram-negative?
Acid-fast is gram-negative.
Acid-fast is.
I'm guessing. Gram-negative.
Why are you guessing if you already know? Acid-fast is only Mycobacterium. Only Mycobacterium is acid-fast. Before you apply this staining technique, you already know it's gram-positive. So here's the question: which one is gram-positive? Acid-fast. That means the red one.
The red.
Yes.
It says pink there.
Whatever. By the way, if you will observe together gram staining and Sill-Nielsen staining, you will observe that redness of acid-fast bacteria is more. It's stronger. It's closer to red, and in the gram staining, it is closer to pink. Why? Even if we use different dyes, they are as red, both of them. The idea is that acid-fast, the ability of acid-resistant bacteria to retain the dye is higher because of this waxy layer.
Okay.
Higher than in gram-negative bacteria. So, when you're asked, gram-positive bacteria, is it red or blue or purple, whatever, you ask by which technique? I say Sill-Nielsen technique. So you say, okay, well, in Sill-Nielsen staining, acid-fast gram-positive bacteria will be red. About others, we can just suppose. Because it doesn't mean that blue ones are gram-negative or gram-positive. We don't care about that.
Just want to differentiate.
It is not the question of this staining technique. The question of this staining technique is which one is acid resistant. That's it. So you know for sure that this one is gram positive. And by shape maybe you can appreciate, oh, this one also is gram positive. But since it is not acid resistant, it absorbed only methylene blue. But here we can just guess. So as long as you get such questions, you should ask some extra, because it can be a tricky question, and you don't pay attention just because you have some prepared answers, but it doesn't work like that. Okay. Also, we have capsule staining. That is also called negative stain. How do you think why it is called negative stain?
Because it's binds to the negative, to the capsule with a negative charge.
Negative charge. I am used with such answer. Huh?
It's wrong.
That's wrong, yes. Negative staining, you can see here. What can you see here?
It stains the medium, but not the bacteria.
Okay, so it's a reverse.
You know what is negative
Yeah.
in photography? All the colors are reversed. It is like there are two ways to highlight something in text. Highlighting the part you need, and highlighting everything except that what you need. In case of highlighting of text, that sounds stupid, right? highlighting the entire page or the entire book in order to highlight one quote. But here, since it is much easier to stain everything around bacteria than staining bacteria itself, capsule itself. Here you can see the capsule. And you can see that structure is pretty similar to that one you saw on the picture before, right? Because closer to, like far away from bacteria, it doesn't have clear delimitation, the capsule. These polysaccharides are going like wool or something. So it doesn't have clear delimitation like the cell wall has. And this is why it is called negative, because you actually apply staining to the background, not to the bacteria or to the capsule. Because actually capsule never absorbs the dye. It is that neutral, it is not able to attach anything. Yep. And for this
If you go down where like the classes or the practices,
So right on the right, the last one on the right. Last one. Go in front. Last one on the right.
So, for negative staining, we can apply Bury method or Bury-Hinz method. When we are talking about Bury method, it is applying only one dye. It is simple staining, India ink. It is black ink that covers the entire surface, and it doesn't cover the bacteria with the capsule. When we are talking about Bury-Hinz staining, after that you apply also the red dye, carbylfuchsin or watery fuchsin. You apply, and that one will stain the bacterial body, not the capsule. So it will penetrate in the bacterial body. As a result, you will see red bacteria, almost transparent capsule, and black background. Or like this. In my opinion, that's enough. But whatever. Endospore staining. Talking about endospore staining, basically talking about any methods of staining, they look similar with different steps. You just choose the chemicals that have more affinity to one or another substance structure. So again, you can highlight spores only by applying classical staining, same gram staining, and so on. And the spore will stay transparent. You will just see the gap inside of the cell, and it stays transparent. Or you can apply malachite green. It is primary stain that has better affinity to spores structure. And after that, it is similar to Sil-Nielsen because we need to apply heating. Like you put the slide above the hot steams. Also, it should be gentle heating. And after that, you wash it out with decolorizer and apply safranin, red dye. So decolorizer will clean only bacteria, and safranin after that will attach only to bacterial cell wall. As a result, you will see difference between spores and bacteria. talking about flagella staining, I like this comparison so much. It should be familiar for girls. Because when you apply gram staining for bacteria with flagellum, you will see only bacteria. You will not see flagellum because they are that tiny and they have different chemical composition of the protein flagellin has different chemical composition from the cell wall. As a result, it will not have such affinity to flagellum. This is why we apply Loeffler staining for detection of flagellum, and this staining includes particles that have affinity to these proteins. As a result, tell me please, how it is called in English?
What?
Things girls are applying to make something looking bigger than it is.
Mascara?
Hmm?
Mascara? I knew it because it kind of looks like eyelashes.
So actually, it doesn't give to eyelashes bigger volume or length. It just covers it with color that makes them to be more visible and to look bigger.
Get darker, bigger.
Same thing here. So it adds extra volume. And as a result, it becomes almost as evident as the bacteria itself. Also, it is not enough knowing only about staining techniques. Different types of microscopy. For an example of bright field and dark field microscopy. That can be made with the same microscope with a small exception. This is a schematical picture of the classical light microscope. And the idea is that we have source of light that goes through the condenser lens. Condenser lens connects the entire light and sends it directly into the smear in order to make the most of the light to intercross inside of the smear. And after that it goes directly into objective and to the eyepiece in order to let you see the picture. So the light passes through the entire smear, and this is why it is called bright field microscopy. Because the entire background is bright and the bacteria is visible, of course, if you apply any staining methods. But not every time we can apply staining. We cannot stain all the time. For example, you need to detect motility of bacteria. There are two ways to detect motility of bacteria, and please pay attention that the form of question is extremely important. When I ask you which is direct method of highlighting of flagellum, direct method, and which is indirect method of highlighting of flagellum. You ask yourself, the word direct refers to what?
When you see the flagellum,
To flagellum. Because otherwise I can ask you which is direct method of highlighting of motility. And which is indirect method of highlighting of motility. And in this case, it will be completely different. For example, when I ask you about detection of flagellum, direct method is Loeffler staining, when you give volume to flagellum, and you can see the flagellum themselves under the microscope. And indirect method, you can see the motility of bacteria. You don't see the flagellum because you didn't apply any staining technique. As soon as you apply staining technique, you attach the bacteria to the surface of the slide, you kill the bacteria, it is not able to move anymore. You will not see any movement. So it will be direct method of highlighting of flagellum, but indirect method of highlighting of movement, because you can see flagellum, you suppose, okay, this bacteria used to move. When we are talking about direct highlighting of motility and indirect highlighting of motility, when you can see the movement, it is direct highlighting of motility. When you can see the flagellum, it is indirect highlighting of motility. When you cannot apply staining technique, but you need to see motility, here we have dark field microscope. We make hanging drop smear. What it means? We have a special slide with a gap. We put here a droplet of saline solution with bacteria inside, potentially motile bacteria, and we turn it upside down for a few reasons. First one, in this way it will evaporate very fast and it will get dry, you will see nothing. In this way, evaporation is going usually up with heat. All the heat is going up and it will not evaporate too fast. And second, superficial extension. It gives more volume to that droplet, and bacteria will have more space to move. So you put this hanging drop smear on the stage. Same thing. We have source of light, we have condenser, but the difference is that here we have central aperture that covers most of light. So it blocks most of light. As a result, it sends the light rays only on edges. Still they intersect here inside of the smear, but most of light is going beyond the objective, not inside the objective. So the light is not coming to you directly. Most of light is going beyond. As a result, the background will be dark, but please don't misuse with the negative staining with black background. This one will be dark. Which is the difference between black and dark?
Black is not.
Black is color, dark is absence of light. Under the table is dark, but not black, you know? So it will not be completely black, it will be dark background. And you will see a little bit of light coming from bacteria. And you will see motility if it is motile. If it is able to move, you will see motility. Why you will see the bacteria? Because the matter, the substance of bacteria that is transparent, differs by density from water or from saline solution in which this bacteria is floating. As soon as light is passing from one substance to another one, the part of the light will refract, the part of the light will go forward. So this refraction, part of this refraction will go here. And you will see this light that passes through the bacteria, you will see it because of this refraction. As a result, without any staining, without any fixation, with this dark field microscopy, you will be able to see the shape and the motility of the pathogen, of the bacteria. There is electron microscope. Please don't misuse with electric. One of the mostly common mistakes. Because nowadays almost every microscope has some electric parts. At least that source of light is electric. When we are talking about electron microscope, it is not like it doesn't run on electricity. It does. But it is not the essential side of this microscope. The essential part of this microscope is the jet of electrons that are passing through the specimen. So we have electron gun is a device that producing these electrons. We have electron beam that sends these electrons. We have anode that change direction. We have magnetic lenses that keep these electrons going forward. We have this electron detector that concentrates jet of electrons towards the specimen. And after that, you can see different types of the transmission electron microscope and scanning electron microscope. So in this case, with scanning, the secondary electron detector that plays role of matrix or screen that receives all the picture will be on the lateral part from the specimen, so it will reflect towards the secondary electron detector. And here a similar thing, but it will the picture will be received on the fluorescent screen. So, what is important to keep in mind? Why electron microscope is that good? Because electrons are the smallest particles we can manipulate, and as a result, they will give us that magnicity, that precision of image. There is no device that is able to show you that high precision of minute details. As a result, what is important? Because probably most of you will not work with my electron microscope, even with any other microscope. I didn't ever approach or touch or see an electron microscope except pictures, Because, by the way, how do you think how much is one light microscope? One light.
The price of one light.
Light microscope.
Euros and they.
Just as you wish. A new one,
1,000.
a good one, not the best one, not the cheapest one.
700 euros. 1,000.
About starting with 1,000 euro and up. It depends on different.
The usual light one.
Yeah.
Light one. Mine
Yes.
is the electron,
Yeah,
mine
the
is
simple.
like tens
Yes.
of thousands.
It can be over 100,000 euros. Yes, over.
Wow.
So sometimes it is cheaper to buy a building in which this microscope will be situated than the microscope itself. So it doesn't make too much sense to apply such expensive devices in clinical practice. Doesn't make sense because why do we need electron microscope? Specifically for viruses. You cannot see virus under the light microscope, you can see bacteria under the light microscope. Of course, any hospital can afford light microscopes. For a simple civilian, it's too much to give 1,000 euro for a microscope, because why? Should I buy it? I don't need it. But for a hospital, it is not too much. While talking about electron microscope, it is too much. Moreover, why should we buy for diagnosis of viral infections electron microscope that is that much expensive, but it is not everything. You need to provide maintenance. And it's not cheaper. So it is important to have it for research, to prove viruses exist, and to find all the structures, to find how to fight with them, to produce everything we need for them, and after that, working with simple stuff like you're going to a pharmacy, you buy express test with 30 lei, and you've got the result of lab investigation in your pocket. No, so it simply doesn't make sense having that expensive devices for daily basis or for clinical research, for clinical investigation. And what you will actually need from here, please keep in mind. The picture given by an electron microscope by default is monochrome, black and white. Why do you need this information? When you're gonna have your finals, you'll have some pictures. That makes me extremely upset on students. Like, you have a picture, it's like a gift for you. It's extra point for you. Black and white picture. Question: which pathogen can you see on this picture? 5 options, just one of them is viral pathogen. You don't need even to know how looks that virus. You already know it's black and white, it is virus. You see 5 options, just one is viral infection. I guess it is hepatitis E or something. Doesn't matter. Your ability of making difference between such things would be much more useful than knowing difference between all the viruses and all the bacteria. You know? Because if you see something that makes you to think, oh, virus, bacteria, that's it. In the real medical practice, you will never need knowing everything about microbiology. But you should be able to make difference between symptoms caused by viral infection and symptoms caused by bacterial infections. It is not that complicated to differentiate them. So all the time you should pay attention to things are extremely important for your future. Not just knowing as much complicated words as you like, or to show you that you know more than others. My advice for you, make your best to find the mostly useful materials for you. Don't waste time for extra stuff, because it could be fun for a while, but after that, not really.
Also, medicine is mostly about specialization in general stuff.
Yeah.
They have a job for general practitioners.
Yes. By the way, as much you will know, as more you will understand, you have no idea about a bunch of stuffs. And as closer you are coming to higher level, as higher you become in your specialty, as easier for you will be say I don't know.
Yeah.
Because and you will understand there is nothing shameful about that. It is quite shameful if you don't know something from your field, of course. But this is the point that a lot of this is how you detect a good or bad specialist. If he's ready to answer everything on every point,
Yeah.
Even if he has idea, even if he or she knows the answer, you know, I have sometimes such students who are coming with some health issues. They say I trust you. I don't trust other doctors. This is why. Okay, but I don't trust myself in this field. I appreciate you trust me, but it is not related to microbiology or even to infectious diseases that is close to microbiology. And I could give you some, even in infectious diseases, I'm not that good because it is about treatment, and I'm not good in treatment. So I can appreciate it somehow. I can diagnose it clinically, but still, it is not my job. So, yeah, that's time. That's time to go to practical class.
Yeah, could you send us the presentation?
Of course.
It's like a 5-minute break.
Of course. On Simu. I'm doing it right now. You have time to move in the class.
Okay.
I send you all the materials, and after that I'm coming.
Can we have a little break?
Sure.
5 minutes is enough.